CRISPR, the abbreviation for Clustered Regularly Interspaced Short Palindromic Repeats, now stands at the forefront of genetic research. In this exploration, influenced by Ann Trapton's 2020 MIT News article, we'll examine CRISPR's intricate timeline. Its origins aren't modern or even human, but rather are nestled in the microscopic world of bacteria, where they evolved mechanisms to combat relentless viral predators. These mechanisms, later discovered and understood by humans, would pivot from the bacterial origins to spearhead a genetics revolution. In the mid-1980s, scientists producing a bacterial genome stumbled upon peculiar repetitive DNA sequences. These mysterious segments, initially found in E. coli, were largely overlooked until the 1990s when Francisco Mojica, a Spanish microbiologist, recognized their potential significance. Mojica hypothesized that these sequences, later termed CRISPR, were more than mere genetic oddities. By the late 1990s, after further research, he proposed that they were part of an adaptive immune system in bacteria, acting as memory imprints of past viral encounters. Through the early 2000s, the function of CRISPR sequences in bacterial defense began to take shape. Scientists observed that when bacteria face viral threats, they will deploy RNA molecules synthesized from these CRISPR regions. Acting like molecular most wanted posters, these RNA molecules guided protein allies, notably Cas proteins, to identify and neutralize the invading viral DNA. The system's elegance, with its capacity for targeted genetic intervention, hinted at potential far-reaching applications. By the 2010s, the biotechnological promise of CRISPR was becoming palpable. Leading the charge were Jennifer Doudna and Emmanuelle Charpentier, who in 2012 demonstrated that the CRISPR-Cas9 system could be re-engineered to target specific DNA sequences across diverse organisms. This finding marked a historical turning point. CRISPR, once an esoteric bacterial defense mechanism, was suddenly thrust onto the global stage, promising precise genetic interventions that previous tools couldn't deliver. As the decade progressed, the CRISPR methodology expanded. Prime editing, base editing, and the discovery of alternative Cas proteins broadened the scope and precision of genome editing. Historically, each of these advancements can be likened to past revolutions in biotechnology. Just as the discovery of the DNA double helix in 1953 opened new vistas, the refinement of CRISPR tools promised to reshape our genetic future. Each refinement carried with it the weight of historical scientific achievements, drawing parallels and lessons from the past. CRISPR, when viewed through the lens of history, stands as a testament to the power of curiosity-driven research. Its roots, deeply embedded in understanding basic bacterial biology, blossom into a tool that challenges our conceptions of genetic health and ethics. As we chart CRISPR's trajectory from bacterial genomes in the 1980s to potential human applications in the 21st century, we are reminded of the intertwined relationship between discovery, application, and responsibility.